In this presentation, I am discussing about the preclinical or non-clinical phase of drug development. The preclinical or non-clinical phase of drug development include the investigation of drug candidate in animal models. It's an important stage in the drug development before translation into clinical trial. According to the definition, preclinical drug development involves the pharmacological as well as toxicological assessment of a new drug candidate in animal models. These studies are conducted in order to establish the safety as well as efficacy of the drug candidate. It can be conducted by both in as well as by in vivo methods, and all these trials are conducted in accordance with the guidelines of good laboratory practices. Usually, cell lines or isolated tissues are used as in vitro models, whereas rodents and non-rodent animals are used for the in vivo testing. For a new drug candidate, it may up to 2 years to get complete. Preclinical drug development involved mainly two categories of studies, that is pharmacological studies as well as toxicological studies. Pharmacological studies include the investigation of pharmacokinetics as well as pharmacodynamics of the new drug candidate in animal models. While the toxicological studies investigate the acute and chronic toxicity nature of the drug, reproductive toxicity, carcinogenicity, genotoxicity, immunotoxicity, as well as toxicokinetic studies. So as we have seen in the previous slide, pharmacological studies include both pharmacokinetics and pharmacodynamics. So coming to the pharmacokinetic studies, it is nothing but the investigation of the ADME, that is absorption, distribution, metabolism, and excretion characteristics of the drug candidate in animal models. Generally, ADME studies are conducted in two species, usually rats and dog, repeated with different dose levels, both in males and females. The major aim of the pharmacokinetic studies is to find out the optimal dose level as well as to provide information about the dose effect relationship. The major parameters investigated under pharmacokinetic studies are area under the curve, T half, Cmax, Tmax, bioavailability, clearance, mean residence time, as well as volume of distribution. The safety pharmacological studies include three categories of studies, that is core battery study, follow-up study, and supplemental studies. In core battery study, the investigator investigate the potential side effect of the drug on organ systems which are important with respect to the life supporting functions such as cardiovascular, respiratory, as well as central nervous system. Then next is the follow-up studies. So it is a continuation of core battery study where a repeated dose is administered in this animal species to get deeper understanding about the potential side effect of the drug in vital organs which are investigated under core battery study. Then supplementary studies include the investigation of side effect on organs which are not addressed in the core battery studies. So the core battery studies include the investigation of the adverse effect of the drug in vital organs, whereas those organs which are not investigated under core battery studies will be studied in supplemental studies. Then coming to the toxicological studies, so the first type is the acute as well as chronic toxicity studies. So in order to assess the acute toxicity, a high single dose of the drug is administered into rodents. So usually male and female animals are included in this study. A single dose is administered at least by two routes, one of which should be the proposed route to be used in the human beings. The animals are observed for any toxic effect or mortality up to 2 weeks. The repeated toxicity study should be carried out in two species and one should be a non-rodent model. Usually, usually small dose of the drug is administered 7 days in a week up to 6 to 9 months to conduct the chronic toxicity or to investigate the chronic toxicity of the drug. So under chronic toxicity, usually 3 doses of the drug should be administered, one high dose, one medium dose, as well as one low dose. The high dose should produce an observable toxicity, whereas the lower dose should not produce any observable toxicity, but it should be comparable to the intended therapeutic dose in human. So under reproductive toxicity studies, the investigator investigate the side effect or potential side effect of the drug in reproductive system. Here the male and female fertility, embryo and fetal death, lactation process, care of young and potential teratogenicity of the drug will be evaluated. Next is genotoxicity, where the investigator investigate whether the drug will cause or induce any type of DNA damage. It can be conducted by both in vitro and in vivo method. The in vitro test is a gene mutation assay in bacteria, and in vivo method is a chromosomal damage using rodent hematopoietic cells. Next is the carcinogenicity study. The long-term carcinogenicity study is carried out particularly if the drug is used for a prolonged period of time. In such type of study, the animal is observed for the development of any type of tumors over a period of 2 years or more than 2 years. Then next is the immunotoxicity study, where the ability of the drug compound to induce immune response or sensitivity is studied. Usually these studies are conducted along with the chronic toxicity studies.